request a resource person to take 40 minutes for her lecture.
Good afternoon, everyone. Honorable dignitaries of the dais, I deem it as a great privilege to introduce the resource person, Dr. Shweta Saran, Professor, School of Life Science, Jawaharlal Nehru University, New Delhi. Indeed, I'm fortunate enough to introduce her because after seeing her profile, I was astonished to see her profile because she has done a lot of contributions to the society. She did her graduation, post-graduation, and also her doctorate from Banaras Hindu University. She started her career as a research associate at Banaras Hindu University in 1990. She headed various positions as postdoctoral fellow, young scientist under DST, research assistant, Wellcome Trust University, UK, then as a leader, the University of Delhi, and now currently as professor in Jawaharlal Nehru University, New Delhi. She has received many awards and honors. I list them: postdoctoral fellowship at Wellcome Trust University, UK, Sonder Forschung Bericht. She worked at the University of Constance, Germany. Swami Pranavananda Science Award for Young Scientists by Indian Society in the year 1992. And she has cleared JRF and SRF CSIR NET in the year 1980. And not only in research, she also has passion towards teaching. She teaches various courses at Jawaharlal Nehru University, New Delhi. Some of them are animal developmental biology, focus for PG students, cell signaling for MPhil and PhD scholars, and she has taught various topics, cell physiology in the Delhi University. Her main areas of research in the field of cell and developmental biology. In her laboratory, she is currently doing her research in autophagic cell death. She has sequenced Dictyostelium genome. And to add further to her cap, she has published more than 30 papers in reputed journals. Not only that, she is also a research supervisor. She has guided 6 students and were awarded doctorates, and 3 were awarded MPhil degrees. Now she is guiding more than 12 PhD candidates. She has received many major and minor research projects. She has completed 10 major research projects funded by various funding agencies such as DBT, CSIR, DST, and ongoing about 7 major research projects. She has given many plenary lectures inside Jawaharlal Nehru University and also outside JNU. She is a member of many scientific board committees, both inside and outside JNU. She has organized many international, national seminars and workshops. She has done a TV program for about 30 minutes, a documentary about herself as a women scientist of India, a series of telecasts in DD Urdu. And by this, I just want to invite her for giving a plenary lecture for the technical session 3. I invite you, ma'am. Thank you.
Thank you so much. Can you see my slides, all of you?
Yes, we can see.
You can see your slides. That's fine. Just a minute. Okay. So the thing is that, first of all, I would like to thank the organizers for asking me to make a presentation here. So thank you very much, all of you. And I would like to also congratulate the department for reaching out a very special year, and congratulations. And I'm sure you all will be going much ahead in time. And very nice of you also. What I understand is today that you all are looking at alternate model systems that can be used. Now, the question is, what do we understand by alternate model systems? I mean, any model system that we would like to study should provide the answers to the questions that we are looking into it. And I think that should be enough to identify. But yes, this is a model system that I'm going to talk about is a non-mammalian model system, if we want to say that. So here I start with Dictyostelium. Dictyostelium discoideum is a model system for cell and developmental biology. And this is comparatively a very new model system, whereby the first paper ever that came out in Dictyostelium was actually in 1935. So not very old compared to the other model system that we just heard about at the moment. Now, this building on the left-hand side is the Jawaharlal Nehru University School of Life Sciences, where I am at the moment. So this is the building that we are here. And this was another photograph that I put, was that in an International Journal of Developmental Biology, which was absolutely dedicated to different aspects of studies in Dictyostelium. So anybody who would be interested in Dictyostelium as a model system, as to what you can do with it, can actually go through this particular issue, which was in 2019 actually, which had come out. And that's amazingly beautifully explained in small amount of explanations, the various aspects of this particular model system. Hold on, something is... Just a minute. Yeah. So to suggest that it's a good model system, an alternate model system that can be used, one actually need to justify two particular criteria. One, does the model organism offer experimental advantages not offered by competing systems? And the second is, will any discoveries made using the model be of wider relevance? And obviously, any model system that we choose to study, it must give me some advantages. And I'm sure absolutely every model cannot be used to answer every kind of questions. But we will discuss here the kind of questions that we can ask in this model system. So this is particularly the life cycle of Dictyostelium, which we have here. So on the left, so this particular organism, I do not know how many of you know about this. This exists actually in two forms. One, it's a vegetative or amoeboid form, and the other is in the multicellular form, that is in the case what is called as a developmental cycle, which is shown here. Now, the thing is that if we are talking about the developmental point here, so during vegetative stage, they are haploid. So the thing is, this organism has many plus points. It is haploid in nature. And when it is haploid in nature, there are many good advantages. There are advantages or there are disadvantages of using this organism, and we'll come across them. But during vegetative phase, what happens is they are surviving by phagocytosing or pinocytosing another in laboratory conditions. And then they multiply by mitotic divisions. So now there are not many mating types which occur in, at least in the discoideum that we are going to be talking about, the laboratory Dictyostelium that is there. Now these, till food is there. So under any natural habitat, if you see anywhere where the temperature is around 22 degrees centigrade and it is moist, they can survive eating all the bacteria and sometimes even dead yeast and all. It can survive by eating that. They can just every 8 hours they can actually divide. So the thing is that they're very happy till food is there. But once the food is depleted, then comes the play of the developmental cycle. What happens there is that you will figure it out that at the time when depletion occurs or starvation occur, it triggers many pathways. And the very starting is the cyclic AMP signaling. And the cyclic AMP signaling, we will come across it later, is very, very complex. And they are the ones under the response of cyclic AMP trigger, they undergo the developmental cycle. Now, the beauty of this developmental cycle is that we know that in most of the metazoans that we know of, the zygote divides by repeated division to give rise to a like an embryo, and then that will grow into an adult. But here, the multicellularity is not by the repeated division of the so-called zygote, but it is by the aggregation of all the single amoeba which is there in that particular niche. So somewhere around 10 to the power of 5 or more can come together in response to cyclic AMP as well as cyclic GMP, and they will aggregate. So that is what is called as an aggregation. So they go on and on until they can make a complete fruiting body. So the terminally differentiated cells are the sorus, which has the spores, and the stalk, which has basically your dead cells. So this is the two types. So here, around 15 to 20% of the cells are destined to die by making stalk cells, and the rest of them survive. So this is something which people would like to understand as to how it happens. Then one which is very important is the migrating slug. So this is the only stage where it can migrate in response, showing chemotaxis, phototaxis, anything, it can migrate to a certain distance, and they show very good pattern formation. And that is what is very important. So here, this is the anterior side and this is the posterior. The anterior is made up of a prerequisite of this stalk and spore that you can see is mirrored by pre-stalk cells and pre-spore cells. So these cells are only, if you talk in terms of developmental biology, no, they are not terminally differentiated. They can revert back to undifferentiated cell types, right? So they will show you what is called as they're just specified cells. So here this is anterior is pre-stalk and posterior is pre-spore. But by the time they become a fruiting body, the anterior is made of spores and the posterior is made up of stalk cells. So this is what is the slug that we talked about at the moment. So anterior is all pre-stalk cells, posterior is pre-spore. In between is dispersed the anterior-like cells, which are actually very similar biochemically to the pre-stalk cells. And there are some different subdivisions of the pre-stalk cells, A, B, and O. We won't go into details of all those, but all I want you to remember is pre-stalk O is closest to the pre-spore region. Now this entire morphogenesis to make the fruiting body, the spores come on top, all these cells will move down. So when they move down, they sort of come down like this, forming the stalk, and this is pulled up. So a lot of calcium signaling goes on apart from the cyclic AMP signaling, whereby the spores are pulled up before they can mature enough. So that is one thing which we are very much interested. Sorry. So, sorry, just a minute. Just a minute. Sorry, I don't know what I did. So the thing is, can you still see my slide?
Yes, sir.
Okay. Yeah. So the thing is, let's just go slowly, whereby we have to figure out what makes Dictyostelium so very important. So if I look at the evolution point of view, just before the diverge, so plant and animal diverged, and just on the same side of the animal, you will find that Dictyostelium diverged. Now this was known as more like the mixed amoeba, so they're more like a evolutionary at a dead end, and the closer ones are the east. But if I look at it at the gene level, Dictyostelium shows more similarity to the humans than to its closest neighbor, that is east. Now this particular figure shows you that with around 100 PFAM proteins that domains of 100 proteins they had taken, and they found that Dictyostelium actually shows similarity both to plants, fungi, and the other metazoans. So they actually belong to the amoebozoa, and they are close to the Ophistochondra that we talk about. Now, the beauty of it is that it shows you characteristics of plants, whereby you can see the SH domain containing protein, SRC domain containing proteins, all these things are there. Plus, if I look at this, the genome, there are 33 genes which show or are orthologs of the human disease genes. Now, if you look at the entire life cycle and the construction of these cells which is placed in them, there are no neurons, there are no brain, there is nothing, no sensory organs that is present here. Then what are these genes doing? How can we utilize them? That is the most important thing that we have to look at. Now, this is a eukaryote. So these are lower eukaryotic system, and that makes it more important. Even if they are amoeboid, they are not cell-based, but these are organismal-based studies that you can do apart from multicellular systems. There only thing is they're very AT rich. So sometimes they can be very problematic for people who are working. Now, the thing is that way back, in 2005, the entire genome was actually sequenced, and that's a cover page of Nature, basically. Now, when we see this got sequenced, what we figured out was that the 10 to 15% of the total population, which are destined to die while making the stalk cell, showed vacuolar cell death phenomena. Now, the thing is, when we started looking, everybody at that time point thought that, yes, it's a developmental cell death and it could be via apoptosis, but there are no apoptotic genes which are found there. What we realized that these vesicles are nothing, but they are, instead of apoptosis, you will find autophagic cell death, which is occurring. And we'll talk about it. So NIH at that time point considered this as a model system which could answer questions raised in autophagic cell death, something similar to what C. elegans gave for apoptosis. So that is how this became very, very important for our studies. Yes, they are very good model system for studying biomedical research, obviously from cell and developmental biology, but you can always extrapolate them. You can study the biology of the human disease genes, which are there, 33 of them, even if you cannot make them as a disease model system per se. Since there are around, this is very gene dense, so the genome size is comparatively small, and the number of genes it has approximately 10,500, very gene dense, and many of them will not even have introns in them. So there are only exons which will be present, and around average you can say is 2.5 kb gene that will be there. Most of the studies, earlier studies, were done on the cell biology phenomenon like cytokinesis, motility, phagocytosis, chemotaxis, and the cyclic AMP signaling. So these were one of the first places where all these aspects were actually studied and then extrapolated onto higher organisms. So that makes this organism very, very important. Now, when we look at it, one of them which I talked about is the slug, where the pattern formation occurs very well. So pattern formation means there is a pattern which is created. To start with, you will find that they are showing you a salt pepper. That means all the pre-stalk, pre-spores are intermingled, but slowly they will show you what is called as an anterior clear pre-stalk and a posterior pre-spore. Now, how do they develop? How do they create such kind of a pattern? And all as a developmental biologist, we know that if a system is heterogeneous, they make use of this heterogeneity to create a pattern. Now, heterogeneity in Dictyostelium has been with the following things. You can have nutrition, you can have cell cycle, you can have calcium. This is just to show you one result with calcium, where so they show you a very clear bimodal distribution pattern. This is dotted ones are showing you bimodal. So you have a large population which shows lower calcium and a small population which shows higher calcium. If I sort these cells and plate them, these are the ones which will form the stalk cells, and these are the ones which will form the spore cells. So they do show a biasness. So there is a predetermination and sorting out during the pattern formation that we see. So there are many for either way you can grow them with glucose, or you can be cell cycle position. They all will show you a biasness in their distribution pattern during this development of Dictyostelium. Now, when the population is homogeneous, it is but obvious that they do not have any heterogeneity present. So how do they create a pattern? So a homogeneous population makes use of talking to the cells amongst itself. So talking basically means we are talking of signaling aspects. So there are many points that can be used to define as to how a pattern is created. And further, I am going to talk a little bit about cyclic AMP as well as diff molecule and tell you as to how patterning is done based on these two aspects. Now, this is just to let you know that when they are growing in the vegetative conditions, they can achieve cell density. So obviously, whenever they have to make a development has to be initiated, when starvation is going to begin, at that time, what they need is how they are going to sense the numbers. So there are huge number of different factors which are present here, which senses the number of Dictyostelium amoeba versus the bacteria on which it is feeding. And that decides as to what would be the size of the aggregate that is being formed. Now, this organism becomes very important as compared to, let's say, yeast, which is also single cell. Yeast has a huge cell wall. So doing biochemistry becomes a little more difficult. But this has a small plasma membrane, very tunable. You can actually work with it. So biochemistry or any biochemical analysis can be easily done by them. Now, in nature, they eat by phagocytosis, whereby they are going to eat what is called as bacteria. And under laboratory conditions, what we have done is that we have created what is called that they can grow in axenic rich media. So you can actually have a clean population of the organism. And there, when we are growing them in a liquid culture, so they are actually undergoing pinocytosis. So both phagocytosis as well as pinocytosis machinery is very well worked out. And actually from here, people have looked into other model systems also. So they are, as they're called, they're born eaters. They are all the time eating. So they can phagocytose almost anything. So if you in the laboratory conditions, if you give them dead bacteria, they are still happy, they will survive. So based on that, now we have a couple of people who are working worldwide. So we have what is called as a dictybase.org. So where you can actually figure out what are the things which are working, the papers, the genome, everything is there. And there is a Dicty Stock Center from where you can get the plasmids, all the different strains that are available. So we are supposed to be actually submitting whatever you are creating. So that's a database that we have here, and anybody can actually get through it. Now, during vegetative cell stage, they are phagocytosing, they are distributed everywhere. But once the starvation sets in, they have to come to common collecting points where the cyclic AMP levels are going to be high. Now, every amoeba has the potential to secrete cyclic AMP as well as to receive cyclic AMP. That's the beauty of the amoeba here. So obviously, if all of them are secreting, all of them are receiving, there'll be a chaotic behavior. So you have an adaptation, de-adaptation system. So in the beginning, the cyclic AMP will be oscillating. So you will have high levels of cyclic AMP, low levels of cyclic AMP. So during that time, obviously, they move step ahead to start the aggregation. So normally what happens, they will first sense where the gradient sensing is. So where is the cyclic AMP which is present there? Then the cell has to polarize itself. So cell polarity is very, very well worked out here. And if anybody interested can actually look into papers here. So since this is chemotaxis, in response to cyclic AMP, what happens is you have to have all the components that are required for detecting cyclic AMP in the anterior end. So when you have a polarity, you will have a leading edge and you have a lagging edge. So the leading edge will definitely have what is called as your actin polymerization, which will be there. And in the posterior end, which is the lagging edge, you will have more of the myosin disassembly, which is occurring. So you can see here, so people have actually, since the genome is known, you can actually do all kind of work here. So people have hooked this, made fusion proteins using reporters like GFP, and you can actually localize them in time as to where they are moving around. And there are obviously, because there is cell to cell, head to head, they can be side to side, all kind of cell addition. They play an important role in detecting the size of the aggregate that is going to be formed. So obviously these are calcium dependent as well as independent cell addition molecules that are present there. So I just wanted you to know a little bit of a history. This is Kenneth Raper, whose first paper was in 1935 and 1940. So there are two papers, the gap of 5 years, and hence after I have not seen any paper of his. So he was the man who actually figured out what slime. So they are called the slime modes. Dictyostelium, discoideum is called slime mode because when it is moving at the slug age, they give you some kind of slimy deposition, which is nothing but cellulose deposits. So that is why the name is called a slime mold as such. So he identified this, and that is all he did. And hence after came John Bonner, who till recently has published papers. So fortunately, I had a lot of interactions with him when he used to visit Bangalore. And most importantly, so actually John Bonner was very interested to figure out a small organism which can be used to study developmental biology. And this is how, so he has a lot of videos which are being uploaded by him. So the very first thing that he showed was that these cells show you chemotaxis. And this is chemotaxis, very taken from book actually. So you can have a small tip filled with cyclic AMP, and if these are wild type, you can see the cells which are coming closer to it, showing you positive or chemotaxis to cyclic AMP. And if they're mutant, you see they will not come. So that was one of the most important things that he figured out, and he found that to start with as an acrosome, and then he realized that it is nothing but cyclic AMP as such. And he looked at the spatial patterning of the pre-stalk, pre-spore. Obviously, there are many papers that he has published. These are two very important things that I just wanted you to know about it. So his experiments did not have any molecular biology in it, but then he initiated a lot more studies with Dictyostelium per se. That is how the group grew. So most of the people were his lineage that you will figure out. Now, this is Loomis from US, and he started more with the molecular biology aspects, and he figured out, so actually he looked at the genetic control of the social amoeba in the during development, and he figured out some of the genes which could be responsible for cell differentiation as such. So Loomis's contribution also was very, very high, and now his group, he's no more, but his group is actually still working. This is where I was trained. So this is Professor Nanjundaya from IISc Bangalore, who trained me in Dictyostelium. So he has done a lot of work on calcium signaling. Even if though he's a theoretist, he has actually done some experimental work on Dictyostelium, and that too on calcium signaling. And this is the work that I showed was actually with him, where we showed that higher calcium level can bias the cells towards the stalk pathway. And that was an important finding for the reason that we all knew that overload of calcium was responsible for bringing out cell death in different organisms. So that's how we figured this out. Now, the multicellularity, as I said, was upon starvation and through cyclic AMP. So this figure just shows you how cyclic AMP can be used outside and within the system. So outside, definitely it is by chemotaxis. And as I said, they are single cells. So calcium signaling here, cyclic AMP signaling is very, very complex here. When I say complex, it means that it is starting from single cells to multicellular. So we are talking of nanomolar to millimolar amounts. Now, in the beginning, when the cells are aggregating, there are nanomolar pulses of cyclic AMP, which can allow them to actually start the aggregation. So that is very, very important. So There are different adenyl cyclases which are important. There are different phosphodiesterases to take care of the levels of cyclic AMP. So this is pretty well worked out. Now, cyclic AMP does not control only the gene expression, chemotaxis, but the inside, that is the intracellular cyclic AMP levels, they are actually very important for spore maturation, and this is taken care of by the adenyl cyclase B or R, as we say, and the spore dormancy is maintained by the ACG or the adenyl cyclase germination specific. So this is how. Now, when we look at the three-dimensional structures, so you have oscillating ones in this region, and this was one of Bonner's student who showed this, and you also have cyclic AMP levels throughout the organism. So you have very high cyclic AMP levels here, and when we say cyclic AMP levels here, they also respond to cyclic AMP comparatively much higher. Remember, when we have multicellular organism, all this will be covered because these are all single cells. So these are covered like put in a bag. And this portion is slightly less or thinner cellulose coating that is present there, which is all actually given out by the pre-stalk cells itself. Now, this just shows you how cyclic AMP is important. So starting from amoeba to fruiting body, cyclic AMP signaling is going on. There are different receptors, cyclic AMP receptors, which are present. So they are high affinity receptors, they are low affinity receptors. They can be from different amounts that they are present there. And that is how the cyclic AMP complex is working out. So there are a couple of papers, one can look into it and figure that out. Now, how do they cooperate in this? So if I look at this, there are two important things. One, there is a cell death, which is developmental cell death, which is occurring. Why should some cells die? How do they know that I have to die? I'm sure nobody wants to die. As Arnab himself said, everybody wants to live. So even they also want to live. So why they are dying? So that's the story will go. So here what happened, all these amoeba, these are all red and green ones, and they feed on bacteria. They make an aggregate, they move around. But you can see there are only some cells which are blue. Blue means that they are stalk cells. So obviously they do not help in the propagation because they're dead cells. And only the red cells are there. So there are a lot of signaling which is going to allow the stalk cells to become dead cells and the spore cells to live. And one molecule which is important is DIF, apart from cyclic AMP, and I'm going to talk about it. But the same principle exists. When you make chimeras with different mutants or the wild types, you will find there are some cheater mutants. Cheater mutants, so there's a whole lot of work which has gone. Cheater means they are going to be important in escaping death and they will live. So that is what is called as altruistic behavior. So there is a whole lot of papers now which has come on the altruistic behavior of Dictyostelium cells, whereby a given population of cells cheat upon the other population to kill them. Obviously, it is very easy to say when we make chimeras with a weaker link, obviously, then they are the ones which will go towards the stop pathway. So that is something which is very common where you can study the chimeras as such. So there are some extracellular secreted molecules, and the two important ones that I'm talking about is cyclic AMP and DIF. So in this case, what will happen is, we know there are throughout its development phase, you will figure it out that they are showing you secreted molecules. And you can find the first one is the quorum sensing, which are glycoproteins basically, which are there. There are chemo attraction, chemo repellent, some other attractions, which you can see is basically cyclic AMP or ammonia. So many things are there, actually you can look into it. And then the entire thing is how during development you can figure this out. So if I look there in between, you can see a lot of these molecules which are important. So there was one which was DIF, which was known, that is differentiation inducing factor, which was known to promote stalk cell. And slowly we have this year, maybe 2 years ago, we found that there is what is called a cyclic di-GMP, which is actually not seen in higher organism. It's more in the lower organism, prokaryotes, that you'll find. They also can promote, so we have a gene for this, they promote what is called as the stalk cell death. So if I now look at it, then if you see that, like movement of cells are very important. Why should a cell move? Even if they do not show you chemotaxis, how are they moving? What are they doing? And that has been correlated more with the cancer studies where the cell migration matters a lot. So what people have done now is they have actually initiated studies where Dictyostelium cells have been used to study the cancer cell migration in them. So obviously migration, we all know from our cell biology that how there is a leading edge, there is a lagging edge. But yes, remember when we talk about cancer, these are normal cells which we are making them cancerous to answer questions. They are not normally becoming cancerous, but they have the property of actually dividing as fast as possible. Now, DIF was the molecule that we talked about, which shows you altruistic behavior. So the only thing is that DIF is a chlorinated molecule. You can see this is a chlorine molecule which is present. We know that there is what is called DIF promotes stalk cell. So people wanted to make, when the sequence was out, people wanted to make what is called as a diffless mutant. But when they made a diffless mutant, they realized that nothing was happening in the phenotype. But once they figured out that these are small metabolites basically, and there are at least 45 genes which are actually required for the synthesis of this. So where you have made a gene disruption is something which will decide as to what kind of a mutant you are. And this work has been done, and this was published by us also. So generally what happens is pre-stock will be wanting to do this. Now, since it promotes proliferate cell death in them. So what people did was they have taken DIF. It is commercially available. So you can use DIF directly, or you can make a compound like Difrasol. People have used to show that they show you anti-cancer properties. So that is how people have looked into a model system in Dictyostelium to suggest cancer cells, as well as to show you that, yes, this is what is called as cell death promotion in DIF. Now, if I look at, okay, so we'll get into slowly with it. So we heard a lot of talk on regeneration also. Dictyostelium also shows regeneration, and Bonner has a series of paper on that. So you can chop the Dictyostelium cells, the slug into as many pieces, and they will make small tiny slugs in them. And then so here there is no growth. Remember, they do not feed once they start their developmental phase. So all the feeding is in the vegetative, hence after they do not feed at all. So they can make tiny little ones, but where they will show you a clear cell pattern. Okay, so the patterning is there, but the size does not grow until and unless you sort of shake them and ask them to feed again, and then ask them to develop again. That's a different issue. So here what happens, why people are looking at de-differentiation. We have heard about de-differentiation at the moment. So generally at the site of amputation, whenever you see, you will like newt and all, there is a de-differentiation where all the differentiated cells will become undifferentiated per se. And when they become undifferentiated, then you will figure out as to how the differentiation program has to be worked out. So in case of Dictyostelium, what you can do is you can show this de-differentiation, whereby you can take any multicellular stages till the slug stage. Hence, after they enter what is called as terminal differentiation program. So then it will not revert back. So till the cells are specified, all these multicellular structures, you can actually just provide them with nutrients and they will start proliferating. So what are the principles which are there? What are the genes which are involved in there? And now people have figured out that there are something, at least one of the histidine kinases out of the 8 that we have is actually important in showing you the de-differentiation program in Dictyostelium. So there are again some papers which are present. Now, as I said that this organism showed you 33 gene homologs from the human disease genes. So you have Huntington, you have Parkinson, you have Lissencephaly, you have so many of them which are there. The question that arises is, if there is no brain, no doubt, there is no sensory organ which is present, then what are you going to do, or why did the genes come here? So Dictyostelium provides you a platform to study the evolution of these genes during evolution of the organism. Plus, you can understand the biology of the disease. They do not make a Huntington disease model because they have nothing to show you. They can show you phenotypes when you are going to disrupt that gene, but then that is not a model system as a disease model system. But then if you want, you can understand the biology, what are its interacting proteins, all these things one can study. And importantly or interestingly is what is this Huntington or any other gene doing here. So people have now made a non-mammalian biomedical model system. So one of the first papers which came out was basically on the valproic acid that people have used. And we will talk about that again a little bit later. So here, like this is just one example that I'm talking about. So there are these blues are all dicty and these are all bacterial. So you can see there are a lot of these genes which are involved in the infection of bacterial pathogens. So a series of papers are there for people who are interested to figure out how Dictyostelium is interacting with different pathogens. Now remember, yeah.
Sorry to interrupt, ma'am.
Hello.
Excuse me, ma'am. Sorry to interrupt.
Yeah, please.
Can you hear us, ma'am?
Yeah, I can hear you now.
I humbly request you to finish the session in 5 minutes, as we have to move to the next technical session.
You want me to finish fast?
Yes, ma'am. Yes, ma'am.
Okay, fine. I'll do it. I'll just do it straight away. Don't worry.
Thank you.
Okay, I didn't know. I thought I had still 45 minutes. So anyway, that is how we'll go ahead with it. This is just to show you about the Huntington. So when the Huntington gene was actually disrupted, so you get different phenotype as compared to the wild type cells. And then when we did these experiments, we realized that this can be attributed to the protein kinases and the cyclic AMP signaling pathway in Dictyostelium. And this is a very nice paper that we have published. One which my lab is very interested in, the autophagic cell death. So if you cut open the fruiting body out here, I'll take 5 minutes. Fruiting body out here, you can see that this is the stalk tube. And the stalk tube is all vacuolated. So the question was then figured out that this is because of autophagic cell death, which is programmed. So Dictyostelium shows you developmental cell death program in them. And this is initiated right at the time of nutrient starvation, which triggers you the mTOR pathway as such. Now, our lab was very much interested in understanding this entire autophagy or the mTOR pathway. And this is just to show you with one mutant cestrin, how we actually study. So you can do microscopically looking at how this they form the autophagosomes form to autolysosomes as such. That is, it's a process. And the same thing you can do by Western analysis to suggest that, yes, the entire process is working or not. Just to tell you a little bit about the diff. So here the pre-spore cell, that is the pre-spore cells, they actually make the poison diff1. And they show you altruistic behavior in the sense that they make diff1, and this diff1 is then transported to the pre-stalk O cells, which are very close to it. And since they have this enzyme dis1 dechlorinase, the diff, which is a chloride molecule, can be actually now treated with the chloride molecule can be removed, and then it becomes poisonous. And these people do not have. So that's how they save. And that is how they are showing you altruistic behavior in Dictyostelium per se. So this is a very good model system to study this altruistic behavior, and people are looking into it. Sex is not very good in this year. They only make what is called as a macrocyst, where they can, and so this is by cannibalism, they will eat all of them. They will make a macrocyst where you will have meiosis undergoing, and then you can have, but they generally have a tendency to revert back to the wild type cells. But recently we have found out that there are some locus for the showing you the segment cages in them, 3 of them which are present there. So I'll just skip some of them here. So this is just to show you the one of the important pathway, which is the endocytic pathway. The endocytic pathway, because they feed all the time, and this is a very collective terminology which is used, endocytic pathway. So the thing is, whatever is eaten has to be processed later on. So endocytosis can be microendocytosis, macro. Micro is basically small pinches they are making, and these are all keratin coated. So the entire plasma membrane is basically very, very dynamic because they are all the time doing this. But when they are undergoing what is called as phagocytosis at that, or pinocytosis, then they will eat. And when these are formed, they can be with some organelles or anything also important structures. When autophagic cell death occurs, this then get transported to the lysosomal. So you have an endo-lysosomal pathway, which becomes very, very important in them. This is just to show autophagic cell death pathway, which is pretty well characterized now in Dictyostelium, and many of you must have read this. And this is the molecule that we are talking about, is the nutrient sensing, and that is dependent on target of rapamycin gene. And in Dictyostelium, we have only one target of rapamycin gene. And when it makes a mTORC1 complex, that is basically with the raptor, and then they promote growth. And when they make it with the Richter, then they will promote what is called as autophagic cell death in them. So there, people are now looking into the mitochondrial biology. And why? Because here the mitochondria is not very reduced. So there are at least 65 genes which are present there. And one have looked into different mitochondrial diseases like the Leigh syndrome and many other neurodegenerative like the Parkinson that people are looking. So that's an outcome of the mitochondria, not because of a gene that is disrupted in the mitochondria. Okay, so one can look into it. These are just to tell you that, yes, they can be used as a pharmacological model system to understand the mechanisms of medicinal drugs. So you can actually now use the single cells to actually test an array of chemicals to figure out and then understand the mechanism, because this is very easily done in a laboratory condition that is there. So these are some. This is the valproic acid, which is actually a Nature paper, and that is important basically in the epileptic and the bipolar diseases. So how Dictyostelium was used to make this as a model system to understand the bipolar diseases, which is actually because of the Wind lithium molecule, and how valproic acid works on it is something which is worthwhile looking into it. And the mechanism that is being used is very similar to what is being used in the mammalian system. And these are some natural products whose mechanisms have now been understood using Dictyostelium per se. And lastly, I would say this is a very good model system which can be used under laboratory conditions to be actually taught to BSc and MSc students. It's a very, very easy model system that can be actually now used. And there, as I said, there is a website whereby you can actually understand and see what all. Otherwise, I'm ready to help. If anybody requires any kind of a help from me, I'm there. So these are all my students who have actually been working. They have gone. Some of them are still working here. And these are some of my collaborations. So I treat Prasanna Nanjundaya as the person who has actually taught me. And these are some of the Indian collaborators that I've had there with me. So I'll thank you all for this. Thank you. Sorry, I didn't know about the time. So thank you very much.
Thank you.
Any questions? Any questions, anybody?
The people who are connected through online can also ask the questions. Is there any questions?
Wait. Ajay, wait. No questions, and thank you so much again for inviting me. Thank you very much.